of T lymphocytes and B lymphocytes in acquired immunity. Recognition of the antigen binding to the antigen. Proliferation and differentiation into effector cells. Elimination of invaders. Provide immunological memory. Recognition of the antigen binding to the antigen and sensitization. Some T lymphocyte, B lymphocyte must first recognize that the foreign antigen present in the body. There are the vast variety of antigen receptors present on different B lymphocyte, T lymphocyte. Only a very small fraction are specific for epitope. Antigen should be present to the B lymphocyte, T lymphocyte until a match is made. Recognition of the antigen occurs through successful match between the epitope of the antigen and antigen receptor on small of B lymphocyte, T lymphocyte. Specific antigen receptor produced by a single T cell, B cell can be identical bind to the same epitope. T cell, B cell can respond to any pathogen, produces molecules containing the same epitope, but B cell, T cell encounters antigen in different ways. T lymphocyte only recognize the fragments of antigenic proteins presented to the cells by special cells called antigen presenting cell, macrophages, dendritic cell, B cell. B lymphocyte can recognize bind to the antigen present in the blood, plasma, lymph, interstitial fluid. Binding of an antigen to the specific antigen receptor result sensitization. Activation of a specific T lymphocyte, B lymphocyte which initiate cell-mediated and antibody-mediated immune response. Proliferation and differentiation into effector cells. Once activated B lymphocyte, T lymphocyte undergoes multiple cell division. Proliferation causes clone. Population of cells that are identical to the original lymphocyte. Some cells of the clones become effector cells, which are short-lived cells that take effect immediately against the antigen to provide primary immune response. Elimination of invaders. Effector forms T lymphocyte are cytotoxic T cell, helper T cell. Cytotoxic T cells use toxic proteins to kill the cells infected with pathogen. Signals from helper T cell activate cytotoxic T cell to kill the infected cell. Signals from the helper T cell activate B lymphocyte to initiate antibody production. Effector forms B lymphocyte are plasma cells. Single activated B lymphocyte forms thousands of identical plasma cells. Plasma cells producing secreting soluble form of the B lymphocyte antigen receptor antibody in large quantities released to the blood lymph. Circulating antibodies can neutralize, inactivate specific toxin pathogen in the body fluids. Provide immunological memory. Following differentiation into effector T cell, cytotoxic T cell, helper T cell are the T lymphocytes in the clones remain as memory T cell, which are long-lived, can give rise to effector T cell if the same antigen is encountered later in the life. Remaining B lymphocytes in the clones are memory B cell, which are long-lived, can give rise to plasma cell if the same antigen is encountered later in the life. Memory T cells, memory B cells can cause strong, more rapid response at subsequent encounter of the same antigen to the body. Immunological memory called secondary immune responses.